Hello, my name is Amber Jolly, and I'm an image analysis scientist within Pharma Assay Services at Advanced Cell Diagnostics, a biotech brand. I want to again remind everyone that you will get the best audio experience today by calling in from your computer and using headphones. We recommend that you do not call in from your telephone. I also want to remind everyone to please mute your microphones. Thank you. Today, we will review the steps required to perform image analysis of ACD's RNAscope and Basescope assay images using the open source image analysis platform CellProfiler. The CellProfiler image analysis project was started in 2003 by Ann Carpenter, Ray Jones, and the Guideline Laboratory. The CellProfiler team is based out of the Carpenter Lab at the Broad Institute of Harvard and MIT. In today's webinar, we will review the RNAscope technology and the general approaches to take when interpreting RNAscope and Basescope assay data, as well as how the scoring guidelines differ between our RNAscope and Basescope platforms. We will review the requirements for image quality before beginning any type of image analysis workflow, and then we will move on to view some sample analysis workflows using the CellProfiler software. Importantly, a tech note will be available shortly on the ACD website, which will contain, which will contain detailed instructions as well. And we encourage you to become very familiar with the contents of that tech note as a good starting point for using CellProfiler. So now let's look at the RNAscope technology. RNAscope is a novel in situ hybridization technology with a unique probe design that allows simultaneous signal amplification and background noise suppression to achieve single molecule detection at a single cell level while preserving tissue morphology. The RNAscope assay works for virtually any genome, any gene, and in any tissue. The technology consists of three major parts: a unique target probe that ACD designs against your sequence of interest, a signal amplification system that amplifies the signal with a high signal-to-noise ratio, and the visualization of single RNA molecules as punctate dots. The RNAscope assay allows for either a fluorescent or a chromogenic labeling, and it is compatible with a variety of sample types, including FFPE and frozen tissue, as well as cell lines. The double ZZ probe design is the key feature of RNAscope technology. The bottom of the Z is a 50 base pair sequence that is complementary to your target sequence of interest, whereas the top of the double ZZ serves as a preamplifier binding site where the signal amplification cascade occurs. A standard RNAscope probe is a pool of 20 ZZ probe oligos that cover a 1 kb region of your target sequence, which is selected based on our proprietary probe design algorithm. Here is a schematic diagram showing the hybridization and the subsequent amplification of the RNAscope assay. Once the double ZZ probe binds, it serves as a foundation and a binding site for the preamplifier molecule. Upon preamplifier binding, a cascade of signal amplification will occur. This is also known as building an amplification tree. If a single Z binds to the target mRNA transcript, the preamplifier cannot bind because it needs both ZZs to provide a binding site. This aspect of the assay helps to ensure the specificity of the probes, and it helps to reduce potential background staining. Here's an overview of the RNAscope workflow. The methodology begins with permeabilizing the sample with a pretreatment kit, followed by probe hybridization and signal amplification described in the previous slide. At the end of the assay, RNA molecules can be visualized under the microscope as punctate dots, which can then be quantified. This last part of quantification is the part that we will be focusing on in today's webinar. Before diving into the image analysis, this slide reviews all of the assays that are currently available from ACD, ranging from chromogenic singleplex and duplex to multiplex fluorescent assays that can detect up to 4 targets simultaneously, as well as the high-plex fluorescent assay with the capacity to detect 12 targets simultaneously, and the Basescope singleplex and duplex assays. Today, we will be discussing in more detail how to quantify both chromogenic and fluorescent signals. It is important to point out that a typical RNAscope or Basescope experimental design includes a few rounds of staining to ensure the quality of the RNA and the tissue being analyzed before moving on to the marker stain. In this example, we run a DAPB probe which targets a soil bacterium, and this image serves as our negative control for the tissue. In the middle panel, we see the same tissue stained using a positive control probe PPIB, otherwise known as cyclophilin B, which typically displays medium expression levels with an expected detection level of 10 to 30 dots per cell in most cell types. The positive control serves as our indication that the overall RNA integrity is good in the sample. Because the negative and positive control images look good, we can consider this tissue QC pass. Then we can proceed to the marker stain, in this case PDL1, which gives us a nice interpretable image shown in the right-hand panel. Now let's review RNAscope assay data interpretation. We frequently get the same questions from our clients, namely, what is the typical RNAscope signal? What does a dot mean? And does the dot size matter? Why do we sometimes see clusters as opposed to dots? We will address all these questions here. In this image, the RNAscope chromogenic red assay was performed, and we see signal, which appears as numerous punctate red dots of varying sizes and intensities, and we can also see some clusters. It is the number of dots and clusters that are important when it comes to signal quantification, rather than the size of the dot or the intensity of the dot. This is because each punctate dot equals 1 mRNA molecule. Both the small dot, which is pointed to by the blue arrow, and the larger round dot, which is pointed to by the yellow arrow, are both considered 1 mRNA molecule. The dot size depends on the number of ZZ probe pairs that bind the target sequence. Sometimes, when the sample RNA quality is compromised and the RNA sequence is fragmented, fewer ZZs are bound, resulting in a smaller dot. But it still detects 1 mRNA molecule regardless of the dot size. Furthermore, a cluster is pointed out by the brown arrow, and this is the result of overlapping signal dots from multiple mRNA molecules. The same concept also applies to the RNAscope multiplex fluorescent assay. One dot represents a single copy of mRNA molecule. This red arrow is pointing to a cluster of individual signal dots. We usually refer to these clusters as transcriptional hotspots. Now let's compare the differences in scoring for RNAscope and Basescope. Our Basescope technology is the next generation in situ hybridization. It is based on the RNAscope technology, but there are several major differences. While the standard RNAscope probe contains 20 ZZ pairs, which target a 1 kb region of the target sequence, the Basescope can detect signal with as low as 1 ZZ pair probe binding. Both the RNAscope and Basescope are capable of single molecule detection, meaning that a single dot represents one single RNA molecule or target. The RNAscope is a robust assay to detect gene expression of most RNA targets, while the Basescope assay is specifically designed for the detection of exon junctions, short target sequences, and point mutations. RNAscope is able to detect up to 3 targets simultaneously, while the Basescope can detect 1 to 2 targets. Detection with RNAscope can be chromogenic or fluorescent, while Basescope is available in the chromogenic format only. RNAscope can be performed manually or using automation, and currently the Basescope singleplex is available on an automated platform. However, the BaseScope duplex is only a manual assay. Please note that the BaseScope probes are designed differently, so they cannot be combined with RNAscope reagents. Similarly, RNAscope probes cannot be combined with BaseScope reagents. Our semi-quantitative scoring is a visual assessment that provides expression levels and the percentage of cells that are positive. We bin the cells according to the number of dots per cell, as indicated in this diagram. With the semi-quantitative approach, you may manually count the number of dots per cell and score the sample from 0 to 4 based on the scoring guidelines shown here. For example, if you see 4 to 9 dots per cell with no or very few clusters, that is given a score of 2. This score is what we would like to see with our positive control PPIB staining on most samples during sample QC. Or if the staining would show more than 15 dots per cell and 10% of the dots are in clusters, that would be given a score of 4. Here are the guidelines for scoring the BaseScope assay. You may notice the BaseScope scoring guidelines are slightly different from the RNAscope assay guidelines in terms of number of dots per cell and the presence of clusters. This is because normally you would expect to see much less or fewer signal dots per cell with the BaseScope assay as compared to the RNAscope assay. The H-score is related to the semi-quantitative scoring method. The difference here is that the bin value is multiplied by the percentage of cells falling into that bin, with the resulting score landing in a range of 0 to 400, where a score of 0 indicates that none of the cells express the marker of interest, while a score of 400 indicates that 100% of the cells fall into bin 4, indicating that all the cells express the marker at the highest possible level. Because data analysis is one of the most frequently asked about topics by our customers, we have several resources available on our website to provide more guidance. One important document that you may want to look at is a reference guide, which was developed by ACD scientists, with examples of different scenarios for RNAscope staining and an explanation of how the data can be analyzed in each scenario. This document is called A Guide for RNAscope Data Analysis and can be found on the ACD website. Before moving on to the CellProfiler analysis workflow, it is essential for us to review the necessary attributes for an image to have before we can be confident in the image enough to move on with analysis. It is very important to be sure you are using the most optimal images for analysis. In order to get the most optimal staining, we always recommend running cell pellet controls provided by ACD to verify the assay setup and confirm the assay workflow when you are starting the assay for the first time. Then running positive PPIB and negative DAP-B control probes to qualify your sample and to determine if the current assay conditions are the most optimal for any target detection. Also, please feel free to contact ACD technical support, and we are more than happy to help with assay optimization. Here are some examples of the challenges we will face in analyzing chromogenic images. The images on the left are the negative and positive control images. We always suggest that you include negative and positive control slides for each tissue as they provide a point of reference. These images on the right will show some examples of false signal. For example, some tissues display a pink haze that is inherent to the tissue and has a similar color to our red assay. But with high quality images, we are able to actually distinguish the colors between this pink haze and the red signal based on their intensity and brightness and slight differences in color, as shown in the adjacent image. There are other structures in the tissue that may have colors. Here we have some examples like tar, lipofuscin, and melanin. It is easier to distinguish these colors from the reddish color, but they may be very difficult to separate from brown signal. So for these types of tissues, we advise against using the brown assay. Importantly, if we start with a bad image data set, we should not expect to get good data out of the analysis. As is the saying in image analysis, garbage in equals garbage out, and you can expect much higher quality data from good quality images. Another challenge in chromogenic image analysis comes from the color quality. Here are two images that are looking at the same region, but they were acquired using two different scanners. As you can see, scanner 1 gives a nice single color dot, while the dots from scanner 2 have multiple colors. The image analysis will be much easier with the single color dots and much more difficult with multiple color dots. So the choice of a scanner and camera or microscope is very important to getting good quality images for the analysis. Next, we will briefly discuss fluorescence imaging. One of the main challenges in fluorescence imaging is autofluorescence. Here you can see the autofluorescence in different tissue types. Autofluorescence is intrinsic to all biological specimens and should be excluded during image analysis. However, this may not always be easy to do, as it may be difficult to separate autofluorescence from rheofluorescent signals. One way to distinguish if a signal comes from autofluorescence is to check if the same signal is present in different channels, because autofluorescence usually has a wide spectrum, which makes it visible through more than one channel. Autofluorescence is exacerbated by tissue fixation and overdigestion in the protease treatment steps of our assay. This checklist is a good way to review whether or not your image is of good enough quality to continue with the analysis. Firstly, check that the colors are well separated for chromogenic images. For fluorescent images, be sure that their fluorescent channel bleed-through is minimized, that autofluorescence is minimized, and the signal is not saturated. Also, be sure that each channel shows the highest dynamic range possible. For all assays, be sure that the images are in focus. And it is always good to have reference images for positive and negative control probes available for each unique tissue and for each unique assay run. The next issue I will discuss is overexposure. Here in the top image, you see the pixels of a fluorescent image. Each pixel has a value, and when we plot those pixel values on a graph, with the pixels on the z-axis, you can see them appear as peaks shown in the graph. What you can notice here is the height difference between the 3 dots in the front and the 2 dots in the back. So here we can actually distinguish between, using the intensity, we can distinguish between the dots in the front and the dots in the back. However, this is an example of what would happen to your image and your image analysis if you were to overexpose that image. When we overexpose the image, all of the dots become very high in their signal, and we actually lose the information that differentiates between the dots in the front and the dots in the back. Not only do we lose that difference in information, but we also see that the size of the signal has expanded on the top. All of this would affect the output of our image analysis. Now let's look at the CellProfiler image analysis workflow. CellProfiler can be downloaded from the CellProfiler website at cellprofiler.org. We highly recommend that you analyze images captured at a 40x magnification or greater and at the highest possible resolution in order to accurately identify these small-ish dots. It is always best to analyze uncompressed images as much as possible when performing image analysis. CellProfiler does have a limited capacity to handle large, high-resolution images. We recommend using a field of view limited to a maximum XY dimension of 2 gigapixels, or approximately 1024 by 1024 pixels, to count cells and punctate dots present within cell boundaries. Many of our customers wish to analyze scans of full tissues, which is a significant hurdle, especially when we are trying to leverage the use of an open source platform such as CellProfiler. One option that you do have is to use this a platform called QPath, Q-U-P-A-T-H. To perform whole slide image analysis using your CellProfiler pipeline, a company called Glencoe sells a plugin to allow you to interface your CellProfiler pipeline with the open source platform QPath in order to perform those whole scan or full tissue slide image analyses. Information on this plugin can be found by contacting Glencoe through their webpage. The plugin works through the Omero Plus platform. There are many different types of software available to you for you to analyze your data. No matter which software or approach you decide to take, the first goal is always to identify the objects in a given image. In our case, this will always be the nuclei or the ish dots. To identify objects, a number of modifications will be made to the original image. This includes separation of channels if the image is chromogenic, removal of background noise, the application of appropriate image processing filters, and the application of segmentation algorithms. Once the objects have been identified, each object will possess a number of metadata associated with it. This includes the object's location, shape, and size. Once we have identified objects, our goal for RNAscope and Basescope data analysis is to count the cells, count the ish dots, and finally to calculate the number of ish dots per cell. The best way to do this is to associate the ish dots or the children objects with their parent object, their cell. Luckily, CellProfiler gives us this ability to associate ish dots with their parent cell and then bin the cells according to our guidelines. For chromogenic assays, the most important step is the first color deconvolution step, to accurately separate nuclei and ish dots according to their color, even though the image itself contains a mixture of all of the colors. For fluorescence image analysis, the channels are already separated, so the first step is not as difficult. However, it is important to be able to distinguish real signal from noise, and this is harder when the pixels differ more by intensity rather than by color, size, and shape. In this example, we have a typical RNAscope image on the top left-hand corner. After color deconvolution has been applied, the image is separated out into nuclear, probe, and background channels. The input parameters used to separate the channels will differ depending both on the software or algorithm used, and also upon the choices made by the individual image analysis scientist, depending on which parameters they decide to use at the time. Therefore, color deconvolution results will vary greatly from user to user and will depend a lot on the quality of the camera or scanner used to acquire the image. With fluorescent images, the channels have already been acquired separately and are ready for individual analysis. Here we see our recommended workflows within CellProfiler that we suggest can be used as a starting point, which you may think of as a backbone or a blueprint for image analysis. The CellProfiler software refers to these as pipelines. The pipeline provides the organizational structure for each analysis and allows a huge amount of flexibility and user-friendliness for all sorts of image analysis applications. Each pipeline is a series of modules which are added by the user. In our demo, we will show you how to create your own pipeline from scratch and to begin to analyze a single-plex chromogenic RNAscope red assay, and how to access a published pipeline from the CellProfiler website to use as a starting point for the analysis of multiplex fluorescent assays. Please do use the tech note associated with this webinar once it's uploaded to the website as a reference to analyze your RNAscope and Basescope data, whether that be a singleplex or duplex assay or a fluorescent assay image. Now let's begin our CellProfiler demo. So just to reiterate, no matter which approach you are taking, whether you're analyzing a chromogenic image or a fluorescent image, it's always important to realize that you will have to tweak every pipeline, the settings in every pipeline, including the thresholding method, etc., to your unique image attributes, because every image is the same. One question that we often get is actually, can I subtract, perhaps using image J, my DAP-B image from my marker stain image? And the answer I usually give is no, because there will be slight differences from one slice to another when you're looking at FFPE tissue, for example. So it's always important to treat every single image as a unique project that needs special attention. Which brings us to the next part of our talk, which is to just let you know that here at ACD, we perform image analysis using the Halo software provided by Indica Labs. The basic principles are the same as what we saw with CellProfiler. The benefit of using Halo software for us is that we can easily analyze those whole tissue scans, but we also have the other capabilities such as automated tissue classification using an AI-based machine learning program, which, for example, can allow us to use that machine learning program to differentiate between the texture of an image so we can separate out tumor versus stroma using the software, for example, rather than hand annotations. We also can perform more complicated analyses such as for ISH, IHC analyses. This is an overview slide of as you can see, the basic nuts and bolts of the Halo software, how it works, and it works, again, quite similarly to what we actually would do with CellProfiler. So all software platforms have a lot in common. The capacity differences are usually related to what size images that platform can handle and the various bells and whistles you can get out of the analysis. The data deliverables from our Halo analysis in pharma assay services include the total cell count in the region of interest, the average probe copies per cell, the percent of cells in each bin for each probe, the H-score for each probe, the percent dual or triple positive and negative, and all of the per cell data provided in an Excel file. We also provide bar graphs of the percent cells positive and H-scores in each region of interest, such as the tumor and stroma, identified using the tissue classifier module. And we can also provide heat maps. With that, I would like to thank everyone for attending today's webinar. Once this webinar is uploaded to the website, you will also have access to that tech note, which I referred to in the demo. I would highly encourage you to go back, now that you've been introduced to the basics of CellProfiler, go back and watch the video again, look through the tech note, and see if you can actually try this yourself. And then please feel free to reach out to support. We are always here to support you. You can email us at support.acd@biotechne.com, and the phone number and websites are listed on this slide as well. My colleagues are still on the line now to help address your questions, so please feel free to continue sending your questions through. Again, as a reminder, if you, in the event that you were unable to get a question sent through the chat feature, I will leave up this contact information for the technical support. And please do stay online a little longer, as we will be sharing some frequently asked questions with you regarding data analysis shortly.